Hello, my name is Melissa Craddock. I'm Assistant Director and Curator at the Bade Museum. I'd like to welcome our online audience to today's installment of the Migration, Borderlands, and Social Boundaries in Antiquity lecture series. Today's speaker is Dr. Megan Perry, who will be presenting a talk called Movers and Shakers, Exploring Migration in the Classical Period Near East Using Bone Chemistry. But before we start today's program, I would first like to invite any audience members who are tuning in live to use the YouTube comments box to post any comments or questions that you may have for our speaker that we will address in a discussion at the end of the presentation. And I would also like to invite my colleague, Associate Curator Jess Johnson, to read a land statement on behalf of the Bade Museum, which is co-sponsoring this talk along with the Archaeological Research Facility at UC Berkeley.
Thank you. We would like to begin by acknowledging that Berkeley, California is on the territory of the Huchun, the ancestral and unceded land of the Chochenyo Ohlone. We respect the land and the people who have stewarded it throughout many generations, and we honor their elders and their ancestors. We are living in a moment that warrants deep reflection on our past and present. As a museum dedicated to advancing knowledge of the archaeology and history of the ancient Levant, the Bade Museum welcomes scholarly discussions across boundaries of nationality, religion, gender, and other aspects of identity. We particularly welcome scholars who are engaging with risky topics in today's fraught political landscape, and whose scholarship bridges past and present through study of issues like migration, borderlands, and social boundaries. Thank you for joining us.
Thank you, Jess. It is now my pleasure to introduce today's speaker, Dr. Megan Perry. Megan Perry is Professor of Anthropology and Rivers Distinguished Professor of International Studies at East Carolina University. She holds degrees from the University of New Mexico, PhD, an MA from Case Western Reserve University, and a BA from Boston University. Her primary research interests involve investigating human skeletal remains to assess ancient disease, diets, and mobility patterns, in addition to mortuary practices of ancient populations from the 1st to 6th centuries CE in Jordan. Her bioarchaeological research at Petra focuses on how one neighborhood in the ancient city adapted to their increasingly urban environment through evidence of physiological stress, isotopic evidence of diet and migration, population demography, and sources of the site's most important resource, water. Professor Perry has been working on archaeological projects in Jordan for over 30 years, and she's on the board of trustees of the American Center of Research in Amman, Jordan. Dr. Perry, the floor is yours.
Thank you so much, Melissa, and thank you everyone for inviting me to give this talk. It's really fun to talk about this. I actually have been talking about other things lately, and so it's nice to kind of go back to my original research in isotopes. So I'm going to share my screen and start up the PowerPoint, and let's make sure everything's going to go right. Yes, there we go. Okay. So I'm going to be talking about looking at migration and population mobility of humans in the classical period Near East through bone chemistry. And to start off, we really want to provide some background in chemistry, right? So we're going to be going a little bit into isotope chemistry. So as you may recall, all elements at the atomic level have a certain number of protons and electrons, as well as neutrons. Isotopes are versions of the element with the same number of protons and electrons, but varied numbers of neutrons, so they have different masses. Take carbon, for example. So you see carbon-12 is one isotope of carbon that is the most prevalent on Earth. So almost 99% of the Earth's carbon is made up of carbon-12, which has 6 protons, 6 neutrons, 6 electrons. However, there are 2 other isotopes of carbon. Carbon-13, which has 7 neutrons, and carbon-14, which has 8. So carbon-13 is stable, meaning it doesn't change in abundance through time, but carbon-14, as a lot of you know, is radioactive and does decay over time with a half-life of about 5,730 years, and this is why it can be used to date organic material from archaeological sites. Basically, calculating how long it has been since that organism was alive based on how much carbon-14 is left in the material. So there are a lot of uses in archaeology of isotopes, not only dating, but also you can look at climatic studies, you can look at diet, not only of animals, but also of humans. And much of my work has focused on the use of isotopes for diet, which I'm not going to really go into here, but also human mobility. So here I'm going to focus on the use of two different types of isotopes, the ratio of strontium-87 and strontium-86, and delta oxygen 18 to understand human migration. So strontium is an alkaline earth element with 4 stable naturally occurring isotopes of different masses. Some of you might have heard of some others that are radioactive, like strontium-90, but those are not natural. They're human-made, basically. So for migration, we use 2 of them, strontium-87 and strontium-86. So strontium-87 is something called radiogenic. So it's produced by the decay of another isotope, rubidium-87, which has a really long half-life of like 49,000 million years. So it's not really good for dating archaeological sites, right? But the ratio of strontium-87, so strontium-87, which is based on the decay of rubidium, and its ratio with strontium-86 will vary based on how long it has been since that bedrock or the bedrock source of sediments and soils have formed. So older sediments, older bedrock will have higher ratio of strontium-87 to strontium-86. There's more strontium-87 because there's been more time for rubidium-87 to decay, creating the strontium-87. And younger bedrock has lower ratios, so basically lower numbers. So to give you an idea of how this varies, right, you can see a map of the US on the left. This is a geological map showing the distribution of bedrock of different ages. And on the right is a strontium isotope map of the US, basically measured from different materials around the country. And some of the older deposits, like from the Precambrian period, you can see here in northern Minnesota, the Appalachians, have some of the highest strontium isotope values. You can see they're kind of in the orange and red here. Whereas areas that have more recent deposits, such as these areas in the upper Northwest, have the lowest strontium isotope numbers, which are kind of in blue here. Excuse me. So how do humans and other vertebrates absorb strontium isotopes in our bones? Well, we get it from a number of sources in our environment. So not only the bedrock and soils, which come to us through the plants that grow in those soils, and then we eat the plants, or we eat the animals that ate the plants, but also from things like dust and precipitation, sea spray, etc. So our bones hold a mix of different strontium reservoirs that exist in the natural environment. Strontium is taken up into bodily tissues after it's consumed, and since it's very similar to calcium in our bones, it readily substitutes for calcium in our bones and our teeth, and that of vertebrates, I should point out. So knowing if someone migrated to a different region during their lifetime is based on comparing the isotopes they absorbed when they were growing up versus those reflecting the region where they were buried, which is presumably where they lived, for a period of time before death. So most of our skeletal tissues, like other body tissues, constantly remodel, right? We have old cells dying to become replaced by new cells. It's sort of turnover, right? And most of our skeletal system, therefore, reflects basically where we spent the last 7 to 10 years before death. But our dental enamel in our teeth forms during childhood and doesn't remodel. So it essentially remains a permanent record of the isotopes absorbed during this period, which what period it is varied from tooth to tooth. You can see here the ages. This is a general schematic diagram, ages of enamel formation in years, and what parts of the tooth form during those periods of time. I usually use the first molar here, which reflects where somebody lived from about the time of birth to approximately 3.5 years of age, when the crown of the first molar, and this is a permanent first molar, an adult tooth, when it stops forming. So establishing what the local value should be at a settlement or a site is a bit more complicated. So a range can be established through sampling materials that either contributed to or contributed to or they absorb similar strontium as the residents of the site. So that would be like plants in a region, right? They would sort of likely reflect the types of the strontium in the plants that would have been eaten in the past. We can look at teeth of small mammals that might be subsisting on like our garbage, for instance. So they're going to be eating the same kinds of things that humans were eating at that site. Or snail shells, which snails are also going to be have a very small home range and are going to be reflecting as the area where their shell actually was forming, which is likely the area where the shell was then eventually found. And I should point out that we use archaeological materials because there are some modern things, soil in a particular region, except for plants. Plants are modern, right? We don't have many archaeological plants. So the strontium of these materials, the strontium isotope values are averaged, and then the range is established by calculating plus or minus 2 standard deviations from that mean. And that's basically how we end up with a local range. And it might not always reflect the entire range of strontium in a region because of sampling issues. So we'll talk about that in a bit. Now, the other isotope ratio that I've used to understand migration is the ratio of oxygen 18 to oxygen 16, which is denoted delta oxygen 18 and is expressed in parts per mil. And there's 2 standards that are generally used to calculate this delta oxygen 18. One of them, the VPDB or the PD Bellamite standard is generally bones have less oxygen 18 than PD Bellamite, so the delta oxygen 18 value is negative. But bones are enriched in oxygen 18. So let me start over. They're enriched in oxygen 18 compared to the standard mean ocean value. So in that case, they are positive. So if you're ever reading isotopic studies that use oxygen isotopes, just know that sometimes they'll show up as being negative, sometimes they're positive, and usually they'll state which, it's general practice to state which standard you're using. And I actually use the PD Bellamite standard. So they're going to be negative. So isotopes originate in environmental water that comes from precipitation, except for ocean water. And that precipitation varies according to climate and topography, or the delta oxygen 18 in precipitation varies. So things like temperature, aridity of the environment, altitude, distance from the coast, how much evaporation has occurred, etc. And essentially the delta oxygen 18 in our skeleton reflects the water sources that we rely on. But these isotopes are pretty tricky to interpret because there are a lot of other things that can affect the delta oxygen 18 in water, such as food preparation or consuming non-human milk, especially the milk of grazers, which is generally more enriched in oxygen 18. So it's going to be a little higher than our local waters. Cooking foods results in an increase of delta oxygen 18. Fermenting, things like adding acid or sugar to grape juice to make wine, for example, increases the delta oxygen 18 in that liquid. If you have a water catchment system that you're relying on, like a cistern or, aqueducts, that kind of thing, that can result in water evaporation. And as a result, it also will increase the delta oxygen 18. So it's really difficult to sort of pinpoint where someone is based on their delta oxygen 18 value, but you could kind of see if somebody has a really different value from the rest of the people at a site. So how do we establish a baseline value? Basically, we can use, there are a lot of maps out there that track modern delta oxygen 18 in surface water or groundwater. We can also take groundwater or precipitation samples and analyze those. But it's important to recall that the delta oxygen 18 of water that's consumed through food or drink gets metabolized, and therefore the skeletal tissue value is not going to be the same as your drinking water value or your consumed value that would be reflected in the water sources that you test. But there are formulae to convert basically your tissue value to a drinking water value to be able to really assess those things. Now, most of the data that I'll talk about here come from bulk samples of enamel. So basically that's taking a chunk of enamel and analyzing it. Because it's a chunk of enamel, it's going to be taking isotopes that were absorbed at different ages, basically at different periods of enamel formation, and you'll end up with an average of those isotopes that were absorbed over the course of years, which is fine for the most part. If you do want very precise data, you can take incremental samples like you've seen here in this, I don't know if it's a sheep or goat tooth. Sorry, Sarah Conzo can probably tell me that. But you can take incremental samples to understand seasonal or annual changes in residence. This is done a lot with herd animals such as sheep or goats to understand like herding practices in the past, how were they moved to different areas seasonally, or checking to see, okay, where did our sheep and goats from our site originate from? Or it could look, can be used as a proxy for environmental change in the past as well. But strontium isotope analysis is kind of expensive compared to other isotopes. It runs about $60 to $70 a sample. So if our question can be answered with bulk samples, we generally do that because otherwise it really adds up when you're doing like 12 incremental samples from one individual. But I've been talking about isotopes to understand migration, but the concept of migration and its sort of impact is largely tied to the concept of identity. And the archaeological study of identity, kind of talking about that in details way beyond the purview of this talk, but just want to reiterate that understanding social and cultural identity or multiple identities from the archaeological record is rather difficult. It's something, archaeologists grapple with all the time. But in isotopic studies, we really need to consider what does it mean to be non-local at a site? Like who would actually be considered a foreigner somewhere? And that may be more than just somebody didn't grow up at a particular settlement, right? So isotopic studies really need to be conducted within the context of the historical and archaeological record. So what do we know about identity in the Greco-Roman Near East? Well, it was a pretty diverse place. Identity was pretty multifaceted and resolved around a combination of the things listed here and more. But one of the things that I always think about is, the people in this region were pretty cosmopolitan. So how would somebody in this diverse group of people be tagged as foreign? Like how different do they have to be? And what elements of that difference are really important? And that's something that, again, archaeologists are still sort of trying to grapple with. And it's important to kind of think about that when we're doing isotopic research in such a diverse area, right? So I'm going to be talking about using bone chemistry to understand mobility and migration in the Greco-Roman Near East within two different contexts. The first, Kirbet Phanon or ancient Phano, is a 4th to 6th century AD settlement associated with copper mines along the edge of the Wadi Araba. And the second study is a group of small agricultural villages in northern Jordan dating from the 1st to 6th centuries. I'll take a drink of water as well. All this talk about oxygen isotopes makes me thirsty. Okay, so the Phano mines are associated with the archaeological site of Phanon, which is located in southwestern Jordan here. This is kind of within the context of the Greco-Roman street system in the area. And it's at the confluence of the Wadis Dana, Gwer, and Shigar, which did provide spring water for residents at the site. But 12 kilometers of the area, 12 kilometers squared, around this site contains over 250 copper mines. So it was really extensively exploited, really from the 5th millennium BC up until at least the 13th century AD. So this is a very active region, and there are a number of sites kind of interspersed throughout this area and on both sides of Wadi Araba as well. Now, a number of cemeteries from different periods are associated with Canaan, including a Byzantine period cemetery, the Southern Cemetery here that dates to about the 4th to 6th centuries. This cemetery was partially excavated in 1996, and there were 45 undisturbed primary burials excavated during that season, but the cemetery is quite large. So we're talking about a small subset of the individuals who were living at this site during this period. Now we know a little bit about Phano from ancient Christian writers. They describe the grim condition for laborers at these mining camps, particularly Phano, which they call out by name. And so Eusebius of Caesarea details the fate of Christian martyrs at Phano during the great persecutions of the early 4th century AD in his Ecclesiastical History and Martyrs of Palestine, where he describes prisoners gouged out eyes, cut Achilles tendons, and shackled and bound legs. And I should point out, we didn't find any evidence of this in the individuals that were analyzed from this excavation. Athanasius, in his History of the Aryans, points to the low life expectancy of Phano miners, that they only lived a few days because the conditions were so rough. Now, reportedly, the Christian martyrs and other prisoners who were sent to this mine came from regions of the Palestine provinces during the Byzantine era, so basically modern Jordan, Israel, Palestine, and also Egypt. We expect there then to be people getting transported over large different distances in order to be live out their lives at working in the Phano mines. So in order to figure out who we have in our cemetery, we have to think about, that there's not only going to be prisoners or martyrs at this mining camp, but they're going to be people like mine overseers, support staff, locals working there for economic reasons, agricultural workers. There are a lot of agricultural fields in this area. So we want to figure out who were these individuals buried in the Phano cemetery. So my colleagues and I analyzed human skeletal material for evidence of not only immigration from another region, but also exposure to heavy metals, particularly lead and copper. If these were prisoners, right, we would expect, based on the evidence that we have, that we would see non-local origin, right, perhaps coming from different regions, many different regions. So there'd be a wide array of strontium and oxygen isotope values in their teeth. And we would expect actually less exposure to heavy metals because they, died from the poor working conditions at the mines soon after arrival, right? If the historical sources are accurate, and their childhood would have been spent in other regions that are not contaminated. And I should point out that even today, the region around Canaan is actually still contaminated from all the mining activity. So if you're staying down at the Royal Society for the Conservation of Nature, Canaan, little, eco-village, just kind of be aware that the soils are still rather contaminated down there. So their local values at the site were based on snail shells and one rodent from the site of Wadi Fidan, which is right next to Canaan. We actually didn't have any animal remains from the graves at Canaan. Just looking, this is a recent isoscape map of strontium isotopes that was created by Wang et al. And just published in 2023. And they incorporate a lot of data, including my own that I'm talking about here, and other studies to create this sort of isoscape, right? So we can see that Canaan is kind of right on the edge of the base of Wadi Araba, which has sort of, a wide array of different deposits of different ages, largely because this is all, wash that's coming down the wadis that lead into Wadi Araba that have materials and sediments coming from a lot of different bedrocks. And running right alongside the eastern side of Wadi Araba is some sandstone, some ridges built in sandstone. Petra, of course, is another site that's located in this sandstone region. So we expect to get some inputs from there. So there's pretty much going to be, and these two areas sort of overlap anyway. So we expect to see sort of a combination of those two. So we compared our, so here's our Canaan values, right? Here is the range of the fauna that we collected at Canaan. This is kind of the broader Wadi Araba range. And you can see that most of the people fall really within those two ranges. And there's a normal distribution. I also always look, because the fauna may not capture the entirety of bioavailable strontium at a site, I also always look at the distribution of the values and see if it's a normal distribution, because generally if there's a normal distribution, there's probably, these people are all probably growing up in the same area. It's only when we see outliers like Grave 102 here that we might begin to suspect that this person grew up somewhere else. So comparing the strontium with oxygen here in this chart on the right, here you can see Grave 102 way off to the right. Their oxygen isotope value is actually very similar to the rest of the people at Canaan, and that's not really that surprising. They might just be living in an area that has a similar, delta oxygen 18 kind of characteristic as Canaan, but the geology is very different. But the range of values seen in delta oxygen 18 at Canaan are not really beyond what you'd expect at people living at the same site and relying on the same water sources. One thing, though, is that the drinking water values after being converted are still way below what we would see what we do see in the humans. They're more like -5, -6. And so what that means really is that obviously people were doing something to the water before they consumed it. So they were either, maybe it was, in a water catchment feature and evaporating, maybe they were cooking, maybe they were making beer and wine, who knows, right? So we definitely know that, that something's happening to the water before they drink it. But in general, what we're not seeing is a real wide array of values, basically. So to better understand what these individuals were doing at Canaan, we looked at copper and lead levels. So in the skeleton, so bioaccumulation of lead and copper would have resulted from residing in contaminated areas surrounding the mines, or through inhalation of metalliferous dusts and flue gases generated by mining activities. And as I mentioned, that toxic environment at Canaan actually exists today. People have tested the soils and found that they still are rather high in lead and copper. So the levels of copper and lead in the ancient burials suggest that about 44% of these were exposed to toxic concentrations of these heavy metals. And this is measured in the micrograms to grams, and the value that it should be before it becomes toxic, is around 1 microgram per gram. And in 10, in some cases, is kind of okay. But you can see that most of these people are above that. Some are really above that. So comparing this to the strontium isotope values, right, we wanted to see if individuals who are maybe non-local had increased or decreased exposure to the mining environment. Of course, we only have one non-local person, this Grave 102 at the end here. And indeed, they do have rather high lead and copper levels in their skeleton. I should point out that the age profile of these individuals and their copper and lead levels didn't seem to have any relationship. So it's not that people who are older, right, had higher lead or copper levels because they'd been exposed longer. So there doesn't seem to be an age-related pattern. Probably what we're seeing here is everyone's kind of a little contaminated because they're living there, but some individuals have more prolonged exposure to lead contaminants that result in their skeletons being very elevated in these levels. Probably, again, like things like working in the mines. Also, if you think about it, engaging in agriculture and stirring up the soil that would have lead and copper in it, and inhaling that might also really increase your copper and lead levels. So I can't rule out that it's people doing other stuff, right? But not everyone at the site had these really, really, really elevated levels. So to me, that kind of points to maybe these were the people working in the mines. Now, who would have been a foreigner at Canaan? We only have one what we call non-local individual in the sample. This is a 30 to 34-year-old male. The closest region that they could have come from would be the western and eastern highlands on either side of Wadi Araba. Now, I should point out that it's really hard to actually pinpoint the origin of someone based on their strontium isotope value, because there are a lot of areas in the Mediterranean, for example, that could have somebody with a value of 0.708301. But we kind of have to look at it within the context of the other archaeological data. And the most parsimonious explanation is they came from nearby, but it doesn't rule out that they could have come from somewhere else, right? Now, would they have been a foreigner? Probably not, but they did probably work in the mines based on carbon and lead levels. The isotope values don't really clarify this, unfortunately, which is always the case, it seems like, with oxygen isotopes. But apparently during the 4th and 6th centuries in Canaan, people from the immediate regions actually worked in these mines, or they were local prisoners, right? They were not brought in from far, far away. And they, it might be that we're not really seeing the Christian martyrs or whomever in this part of the cemetery, if in fact they did experience the type of life that's explained in the historical sources. So the other example that I want to talk about is our small agricultural villages in northern Jordan. So this past year, a Jordanian colleague of mine had a Fulbright fellowship to be here at ECU. And we worked on a project assessing mobility between the small agricultural villages in Jordan. So this is a period that sees a high population density. You can kind of see on the picture on the left here where these are all Roman and Byzantine agricultural sites in northern Jordan. So kind of here, the Golan or Jolan's up here, and then, the West Bank and Israel's over here. So this period, as I mentioned, saw high population density, and probably because the stable climate facilitated extensive agricultural production. This is a very agriculturally rich area. You can see this picture, that shows the site of Yasile, one of the sites we looked at. This Yasile is located right about here, but the entire region is surrounded by extensive agricultural fields that look pretty brown in this photograph that I think was taken in August, so really at the end of the dry season. But if you came in the winter or spring, these fields are extremely green, extremely verdant, with grains growing, and you can see there are orchards and that kind of thing as well in this area. So we know that the urban cultural centers in northern Jordan, places like Jerash or Qais, they drew a lot of non-local individuals into the site. But what about these smaller agricultural villages that were linked economically and administratively to the larger urban centers? We see population growth in these sites. Is that due to people moving in from outside? So one of the issues, of course, is that the increased size and density of these communities could leave them vulnerable to climatic changes, such as the late antique Little Ice Age. This was likely due to repeated volcanic eruptions that impacted the global climate during the late 6th, early 7th centuries. So we looked at evidence from 4 different sites in northern Jordan: Natfia, if you look here, Yasile, Yamun, and Saad, that are the cemeteries were used leading up to this climate crisis to see how population mobility may have shifted and to better understand the economic relationships with each other, and also nearby urban sites. So these agricultural communities, we know, were largely self-sufficient and practiced subsistence agriculture, but it's not impossible that trade of certain crops occurred between them. We have really large wine presses like this one at Yasile that point to extensive wine production, probably for export. We do have churches with elaborate mosaics at all of the sites except for Natvia, which points to local prosperity. So these sites seem to be doing pretty well during the Roman and Byzantine period. One of the tricky elements, though, of assessing mobility in this region is the homogeneity we see in the bedrock and the soils. We might expect to see an intrasite difference due to things like maybe soils were being grown in crops were being grown in slightly different soils, or the sheep and goats that people were eating at a particular site were moving in a slightly different pattern across different geological substrates, right? So we might see some variation. In general, though, you can see that the area of northern Jordan is pretty homogeneous with these sort of Cretaceous limestone deposits. So we based local bioavailable strontium at these 4 sites on a combination of rodent samples that I collected and Abdullah, my colleague, collected, and data from plants and snail shells that were collected by a larger isoscape study at Durham University in the UK. And the project PI kindly provided those data on these particular sites for us. So what do we see? We do indeed see small variations in the local ranges, which are represented by these shaded boxes. They not only differ in terms of how wide the range is, but also kind of where their mean would be located. Now, even though these sites are in similar geographic and geological areas, right, they're probably reflecting different impacts on local strontium. All of the sites show potential non-locals, people kind of sitting outside of these local values. But we don't really, well, there are a couple of outliers, this one here, perhaps these 2 individuals there, and this individual here, even though they're still in the range. We can't really conclude that these individuals are true non-locals, and that's because they still fall within the range of northern Jordan limestones, which is the box here that has the little crosses in, the plus signs. I also plotted on here the range of basalts, the basalt fields that are located just to the east of these sites that have, because they're volcanic, they're newer, so they tend to have lower values, strontium isotope values, just to kind of see, do we see an impact from this different geological region. And the site of Yasile does show, a little bit lower on average human values. It could be, this is a later site. It could be greater inputs into the diet from the basalt region to the north and east. They are kind of closest to this region compared to the other sites, but nothing suggests that, that we really do have individuals living from far away and moving into these sites. But we do want to understand something about where they're getting their food from. So some other people have been doing carbon and nitrogen isotope studies of the individuals from these sites. So I apologize for the quality of this graph. This is the only one I've been able to get. It's a master's thesis at Arkansas, so it's pre-digital, right? So, but the carbon, I'm not really going to go into the isotopic investigations of diet. Just trust me when I say that the diet did vary within the sites. Most people are eating things like cereal grains, wheat and barley, cereal grains like wheat and barley, fruits like grapes and figs, olives, right? This typical Mediterranean diet versus millet, which is a plant that's grown more in arid regions. It's often kind of relied on in times of maybe climate crisis, for example. So there's some variation in terms of millet or plants like that versus these other more typically consumed plants, but it's not that much. They're also definitely consuming meat and animal byproducts like milk and yogurt, and at different levels, right? So some people are eating a lot, some people are not eating as much. We also see some intrasite variability in terms of, I'm sorry, not intrasite, temporal variation over time, right? In the herding practices of different animals. This was another study that was published in 2015 that shows the difference between the Bronze Age and the Byzantine. Basically, these are Byzantine sheep and goats, Byzantine cattle, and late Bronze Age sheep and goats in the orange. And what this graph suggests is a shift in dietary sources and climate from more arid regions in the Bronze Age to less arid regions in the Byzantine period. So some of this we can propose that some of this intrasite variability in the humans could be due to seasonal or temporal variation in these sheep-goat herding patterns, that leads those animals to rely on plants that might grow better in arid regions at different times of the year. And this is where the incremental analysis of sheep and goat teeth would be really crucial to understand the sort of herding practices, and then how is this affecting not only the diet of these individuals, but also because, of course, they're eating the animals that ate these plants, but then also the strontium isotope values of these individuals. Perhaps that is causing some of them to be sort of out of bounds. Now, we don't really have evidence of interregional mobility at these sites. We don't really have strong evidence of interregional mobility, but we can't really rule it out either. The variation that we see is probably temporal and maybe due to differences in dietary sources and herding practices, rather than people really moving in from somewhere else. In general, these sites were really resilient in the face of climate change. It's kind of surprising. So they remained occupied into the Islamic period, so after the late Antique Little Ice Age. Now, the question is, even if we do have interregional mobility, would somebody moving from one of these sites, say Yasile, to a site like Saad, be considered a foreigner? And that's something that's not really clear. It would be interesting at some point to be able to assess, patrolocal or matrilocal marriage practices. You could see if, for example, women versus men have really wide, a wide array of strontium isotope values. If the women are really varied and the men are all kind of the same, that would suggest maybe the women were coming in from elsewhere. And I mean, we can presume intermarrying with individuals in that community. They could always be coming in for other reasons too. It's important to keep that in mind. But the problem with these sites is that we have a lot of loose teeth and a lot of commingled bones. So we can't really, unless we do something like proteomics, can't really get at the sex of these individuals. So in order to do that, kind of sex-based differences it would be very difficult. Okay. So I want to thank Dr. Brody and his colleagues from the Bode Museum, and also people from Berkeley, for organizing this session and inviting me to give a talk. And then, of course, I want to thank all my colleagues, including the Fulbright Scholar Program, who facilitated Abdullah Sharman's time here at ECU to work on these the strontium isotope studies of Northern Jordan. So I'm going to stop sharing, and that's it.
Thank you so much, Dr. Perry, for a really exciting and stimulating talk. I have a whole list of questions ready for you. I also want to remind the YouTube audience to post any questions or comments that you might have about the talk, and we will address those. So while people gather their thoughts, I will get us started with a couple of science questions. I'm more of a humanist when it comes to how I approach archaeology. So, um, if you don't mind, um, you did a great job of explaining a lot of the basic science, but I have a few questions that might be kind of simple for people who do isotopic analyses on a regular basis. So, the first question is about the level of granularity of regional identification. So given the homogeneity in the bedrock and the soils that you mentioned in regions like the highlands of northern Jordan, what is the level of regional granularity that can be identified through isotopic analysis?
Well, that is the big issue, isn't it? And most of the variation geologically in Jordan is and Israel is east to west, right? So if somebody's moving from east to west, you're going to be able to identify them more likely as a non-local than if somebody's moving from north to south within one of those bands of geological similarity, I guess you could say. And so this is always the question. This is why I wanted to bring in oxygen isotopes, which really can help because we do see people, who grew up on the Mediterranean coast, for example, having very different values than, somebody growing up at Canaan, for example. At Canaan, it wasn't, well, I mean, it's kind of hard to see because we don't really have really non-local individuals, we don't think. But the oxygen would be sort of another way to look at this. There are other isotopes we can look at as well. There's lead, which does vary also sort of based on the geological substrate, but it can, especially as we get into the Roman and Byzantine area, it can reflect if people are using lead or have lead in their water pipes or whatever, that it reflects where the lead is being smelted. So that's one issue. And actually we're kind of trying to look at sourcing of lead across the Roman Empire. It's a project someone at Durham, Janet Montgomery, is working on, and it should be pretty cool. There's also sulfur, which is something that I've been trying to get into to try to get a sense of how it varies across the region, to see if that would be another alternative solution to understanding migration. And there's the old ways, right? Like looking at, what are they buried with and, stuff like that. So it's not totally divorced from any of that.
Right, right. I have several follow-up questions that relate to a lot of issues that you just raised, but I'll first go to the one about lead and heavy metals, because that is an important part of your study of these populations from Finon. I'm wondering what some of the health impacts are from these metal contaminants, and if you see any skeletal pathologies in the Finon individuals that might relate to heavy metal toxicity from their exposure to the lead and copper from living near the mines and
That's
working
a
on the
really
mines.
good question. I didn't. You might expect to see some. I mean, it could be things like not like anemia or something like that. That could be showing up in individuals that have high lead levels. But a lot of the things that might be showing up are, having to do with like your heart and your lungs, and that's not really going to be reflected on the skeletal remains. One thing actually I haven't done and I'd like to go back and do is look at respiratory disease and see how, because of course working in the mines would probably make you more prone to that. And that would be another indicator, right, of what they were doing and kind of the health impacts of that. I mean, we can assume that it wasn't great health-wise, but weirdly, the only thing they kind of showed in their skeleton was a lot of arthritis. And not even anything more than what you'd see at like an agricultural site like Rehovot in the Negev. So it wasn't like they were toiling. These people anyway, these people are not toiling in the mines for their whole life.
Okay, interesting.
Yeah, enough to get toxic, but
Right. But not enough of a habitual heavy labor for that to show up in any clearly patterned ways compared with other ancient populations.
Oh.
Okay, so now a methodological question that relates to your comment about using other kinds of archaeological evidence to help identify locality, so local, non-local, and mobility patterns throughout life from human burials. And I want to dig in a little bit to your comment about being cautious about not conflating isotopic results about locality of an individual or even a population group with cultural identity, because that touches on some really important issues in analysis of human burials and even in archaeology more broadly. And
Mm-hmm.
I think that's also a critique that can be applied to other categories of evidence when they're used alone, like ceramics, the classic pots equals people issue,
DNA?
and also ADNA.
Yeah, it's people, yeah. It's,
So yeah, so I'm wondering how your work relates to this methodological issue in general, and also if you have used ADNA in your research and the way in which it relates to your isotopic
no.
evidence.
I mean, there are some contexts where I think ancient DNA would be really helpful, particularly at Petra, actually. Um, but a lot of one of the reasons why I've actually been contacted a lot to provide samples for some of these larger ADNA studies, and I've kind of resisted because of the way the data are interpreted, and it's getting better, and there are some people who are doing a really good job, but a lot of it is sort of creating these typological identities based on DNA that don't really have anything to do with what might have been their own positive, I mean, their own personal identity, right? And so it's kind of DNA sort of has primacy in that case, and it's kind of ignoring a lot of the archaeological data that we might have on this. And so I've kind of, yeah, not wanted to get involved in those kinds of studies, um, or at least, be responsible for handing over samples for those kinds of studies. So, yeah, so I think it's a really important consideration. And it's funny because I was recently thinking about this a lot in terms of like Nabatean identity with Petra. And so, how did people have a Nabatean identity? People are living within the Nabatean kingdom, and what did that mean? And what's interesting is that like people never actually in inscriptions or anything referred to themselves as Nabateans until after Roman annexation, and only people that were outsiders talking about them referred to them as Nabateans. So did they actually consider themselves to be part of this cultural sphere, that this unified cultural sphere, or did they have their own identities, kind of like the list I showed of like tribal affiliation, and are they nomadic, and that kind of stuff. So it's something that is really, it's critical to think about and try to draw in as much evidence as possible to really understand. And it's also, if somebody is showing up isotopically as non-local, for example, we have someone at Petra who is this older woman, and she's buried along with everybody else and incorporated in the mortuary record in the same way. And so does that show that maybe she really wasn't considered an outsider, right, at the time of her death? So it kind of adds, if you turn it on its head, it kind of adds more data to that understanding of identity.
Yeah, thank you. And you raised so many important points about not oversimplifying ancient identity and the many unknowns and complications and um, those issues of over-reliance on any particular set of evidence. So that brings me to my next question. As someone who also studies human burials, I'm often looking at multiple data sets, including the material culture and burial assemblages. Could you talk a little bit about what kind of patterns you've seen in these burials in terms of the assemblages that these individuals were buried with, and if there were major differences that map on to any of the locality issues that you've identified?
Yeah, no problem. I don't have those data for the Northern Jordan stuff, but for Feinan, and it's been a while since, I mean, I kind of actually read over, there's just one site report that was published in Levant on this. Otherwise, it hasn't really been published that much, and except for what I've done. And so, from what I remember, there were, excuse me, two burials that were really ornate, uniquely ornate in terms of the burial goods that were interred with these individuals. And they didn't have an isotope value that was any different than anyone else. There was one grave, so most of the graves are just sort of, cyst graves dug into the soil. There was one burial receptacle that was like built on the surface, and the individuals from there as well. And now I can't remember if they're the same individuals that also had the lots of grave goods. I can't remember off the top of my head. But they also, I remember looking at that very closely to see, do they look any different in terms of any of their sort of skeletal data? And we don't really see that. So, but it is an important factor, right, to consider. In a lot of other studies looking at migration, one of the reasons why they go and, do this type of research with isotopic data is because they might have some burials that, are really different from others. And they want to see, are these people that are maybe not from around here, right? So it definitely is important.
Hey, thank you so much. Well, we're coming up to the end of the program hour here. So if Jess is around, yes, okay, I'd like to, well, first of all, thank our speaker, Dr. Megan Perry, for this incredibly interesting talk. As you can see, it stimulated a lot of questions for me, and I'm going to keep thinking about it for the rest of the day and look up a whole bunch of the references that you shared. So Jess is going to remind our audience about the upcoming talk in this series that will be in January.
All right, thank you so much, Melissa, and thank you to everyone. Please join us in warmly thanking today's speaker, also Dr. Megan Perry, for sharing her research. It was an excellent talk. The next talk in our series is actually a rescheduled one. We had to reschedule due to some technical difficulties last time. So it will be on January 9th at 9:30 a.m. Pacific time. Dr. Ann Killebrew will be sharing a talk titled Early Israel and the Philistines, the Materiality of Migrants, Refugees, and Colonizers at the End of the Bronze Age. So with that, we look forward to